Start. I've had this mess with me before where something like that happened. It just didn't record. So I'll either have, it'll all be fine, or I'll have two, one shorter and one longer recording. Either way. Anyway, so back to sponges, right? 3 layers. Epithelium on the outside. The choanocyte layer on the inside. The choanocytes, remember, in the purple that you see, were those things that look like those protozoans. Remember I showed you the choanoflagellates, which are protozoans? And my gosh, they looked almost like they could be brothers, right? So again, that idea of protozoans being the base, because I showed you some protozoans that looked kind of like fungi, and some protozoans that kind of look like plants. And now we looked at some protozoans that looked kind of like animals. So you see kind of what's going on with the protozoans. You know, they diversify. So come out of the archaeans, come out of the prokaryotes, and they begin to diversify into a bunch of different kind of weird, some kind of not so weird, but some kind of weird. And then from there, the fungi, the plants, and the animals all come out of, right? And it looks like what happens, at least in this particular case, is those protozoans begin to stick together. Remember I showed you in the last chapter, not just stick, here, communicate, specialize. In other words, they became tissues. Now, they're not tissues here, but these little choanoflagellates, we would believe, were the rise of these choanocytes, these little cells that are lined on the inside, and they have the flagella to move the water. And, of course, if you look at that, then they must be filter feeders, right? We've talked about that. Either, you know, the water moves by you or you move the water by you. So you kick those flagella back and forth, and those collars, that those things sticking out called the collar, they with mucus, how nice, right? They grab that stuff. They grab that food as it floats through. So that's why the big openness we saw on the slide made sense, right? You don't want to be real compacted as the sponge. You want to be kind of loosey-goosey, right? And if you're diffusing and going to exchanging your gases, that also helps with that too, right? Because there's a lot of impenetrable holes that gases can diffuse, because these things obviously, you know, respire. So there's the oxygen and carbon dioxide thing going on. The middle layer, all right, now that is what they call a gelatinous matrix. So imagine that is goo. Yeah, I don't know if that's a good word for it. But anyway, within that soft, gooey material, this gelatinous matrix, are these things that we saw under the scope. And there's some electron microscope pictures of them. They're called spicules, remember? They could be made of calcium carbonate. I think it's siliceous is how you say the word. But in other words, they're made of glass material. There are actually sponges out there. You might have heard them called glass sponges. I'm getting ready to spell glass, for goodness sakes. Have you guys ever heard of glass sponges? The spicules on those are made of that glass-like material. And even in the sponges in class, you know, they were rough, right? You wouldn't want to, I guess they'd be good if you were trying to scrub off skin, you know. Yeah, glass sponges, if you feel it, it almost feels like little bits of ground glass. It's really kind of interesting. But they use those in different shapes, different materials, and as I said in class, folks who know all about this actually can look at some spicules, or maybe all of them, I don't know, and tell you what species they came from. So that's species specific too. Isn't that neat? Mixed amongst all of that is spongin. Think of spongin as a protein. So again, we've got this stretchy kind of material. So with a gelatinous if it was completely just gelatinous, it would just go apart, right? So you take the gelatinous material, put in the spicules, add the protein, and you get something that kind of has some integrity to it, some structural integrity. It's still this loose thing of cells, but it's kind of floating in this sea of gelatinous matrix and proteins, and I think you guys are getting what I'm trying to talk about there. I mentioned in class the other day that they can reproduce by this. This kind of demonstrates their ancestral nature. They're more protozoan-ish, closer than us anyway, type nature. When you fragment them, you could scrape them. You could take them on a cheese grater, a live one, and scrape it, and the little things will go back in the sponges. It's called fragmentation. It helps if you spend your life living on the seafloor, especially if you're in the tidal zone, you know, where a lot of sponges are. Not those deep ones, obviously. But it helps, it helps disperse with anybody. But if you're getting pounded a lot and getting broken up, those little pieces will become other sponges. You can read through that next part very quick. Most hermaphroditic, in other words, they have male and female organs. But look at what happens, how reproduction occurs. Choanocytes become sperm, then go out through the outcurrent hole at the top. It's called the osculum. Water comes in the sides, remember? Follow the blue arrows out through the top. So some of those choanocytes become sperm, go out through there. They drift around, most of them dying and being eaten and never finding a home, but some of them actually being taken up by another sponge and then passed to an egg cell sitting there in that spongy thing, that mesohyle, which sounds like I'm saying something in Hebrew. The mesohyle is where the gelatinous matrix is. Anyway, there's an egg cell in there. And what's cool about this, and I underline it, is there's a bit of development that occurs within the mother in some species, within the mother. So the mother, already there's this idea of maybe, try this out, life. Instead of having lots of offspring, maybe have fewer. Now, sponges still have a lot, or producing lots of gametes. Let's start with that. How about we make fewer offspring and let some of them get a better chance by developing inside of the mom? Have you seen this before? Right? Have you seen this before, this idea of a starter pack, if you will? And what critter did you see it in? If my dog gets too loud, please tell me. Yeah, but we didn't talk about mammals yet. How about a critter that we've talked about? An organism, a type of living thing that we've talked about before. That it's not even an animal. Not an animal. It wasn't an animal. It was a non-animal organism that did kind of the same thing, that sort of decided to give its young a little start, a little chance, not just throw it out there willy-nilly to the wind, but say, how about we take our young and maybe put a little bit of, I don't want to give the word away, all right, endosperm. You remember that? That little bit of endosperm that we might add to the developing young to give them a starter packet? You recall me saying all that, right? Who did that? Which group of organisms that we were talking about started doing that? You gotta be able to hear the dog. You don't really remember? You don't remember how the angiosperms, you don't remember how we talked about triple fertilization, and we talked about the fact that one of the reasons that happens is because the little developing seed, the little junior plant, has a little starter pack to start with. You remember a corn kernel, when you cut it open, only a little tiny corner of that is junior. Most of that yellow stuff is endosperm, food. All right, so the young gets a little bit of a starter pack. So we're giving the young a little bit of a head start. The same thing's kind of happening here. The sponges aren't giving the young a starter pack of food, right? But they're increasing the probability that that organism is going to survive by giving it just a little bit of time to develop inside the mother. Protection from predators and all that kind of stuff. Now, if you're out in open water, well, then fair game. What are you going to do? Okay, so that leaves behind the sponges and moves us now to our first group of critters that actually had tissues, two sets of tissues, the ecto and the endoderm. And those are both of these, the C's are silent, by the way, the ctenophorans and the cnidarians. And then they were eumetazoa. Eumetazoa meaning literally true tissues. So we start to see organisms that aren't parazoa. Parazoa means tissue-like, tissue-adjacent. Eumetazoa, U means true tissues. So two sets of tissues in the ctenophorans, ones we'll talk about first very quickly, and then a bigger group, the cnidarians, which if you live in Florida, we're very familiar with the cnidarians. Ctenophorans are some of my favorite, though. Now, in spite of the fact that the way it's drawn here, we're not exactly sure. We used to put the ctenophorans in with the cnidarians, the jellyfish, right? These are, if you're thinking, what the hell is a ctenophorans? Comb jellies, C-O-M-B. We used to put those in with the jellyfish, but they don't anymore. So down here, maybe there might be more of those dashed lines than straight lines. The phylogeny is not exactly clear as to where they came from. As I said, we call these the comb jellies. Sea walnuts, sea gooseberries. They look like jellies, which is why we put them in the group with the jellyfish, but that could simply be just because of a lot of convergent evolution, or maybe they're related. They have tentacles, but they don't sting you. You see, that's the big thing. The next group, the cnidarians we're going to talk about, they all have one thing in common, and that is they have a stinging cell. These guys don't. All right, they have these fused cilia, and they beat in this coordinated fashion, and they have these tentacles, right, that they're called coloplasts, that don't sting their prey, but stick to it. So, the question is this a jellyfish that's decided to go away from stinging to sticking, or is this a critter that came around the other route and said, hey, I'll capture prey by sticking to them? You know what I mean? It could be either way. That's why the phylogeny, in the end, we say it's unclear. That's an interesting question, don't you think? Is this a jellyfish that said, I'm going to go out on this path? Or is this a critter that was over here doing its thing and then just kind of ended up saying, hey, this is a good way to catch prey, but it just so happened it does it with the glue than it does it with the thing I'll talk about in the cnidarians. How neat, huh? Now, what's really cool about these, the fact these are the glow-in-the-dark things. You probably have seen videos about them. I bet you guys have. There's a there are bioluminescent bacteria that live in these comb-like plates on the side, right? And whenever these plates beat back and forth, whenever these bacteria get agitated, they glow. It could be a predator defense, for example, like if a predator is coming after these bacteria. Of course, we're talking at a really microscopic level. And they all start glowing. Maybe there's a blinding effect or, a distraction event. So when these fused cilia plates beat back and forth, it agitates those bacteria and they begin to bioluminesce. Isn't that neat? I used to see that in the Navy when I was out on the ship at night as it went through the sea, as it cut that V through the water, especially if it was nice and calm and we were floating in a bunch. We would just be in some of these bacteria, plankton, do this as well. And it would be a green line that would go. It spread out from the ship. It's absolutely incredible. If you haven't seen some of the videos, go on YouTube, find some videos of comb jellies. It's beautiful. They and when these fused cilia beat, like they do it kind of. I don't know how to explain that. They kind of move down like cards do. The colors change. It'll go red, green, yellow, blue. Red, green, yellow, blue. It's absolutely incredible. One of the neatest critters on the planet. But where they came from, nobody quite knows. That could be your. That could be your goal in life. That could be your destiny. Find out the phylogeny of the ctenophorans. Okay, but now the ones that we truly call the jellyfish, the ones, like I said, living in Florida, we're all very, very familiar with, and that's because they sting you. Okay, there are a few freshwater species, for example, hydra, which you're probably more familiar with than you might be with any of the other ones. That's a freshwater species, that hydra that we looked at on our microscope. But they're all carnivorous. They're all carnivorous. They're all radially symmetrical. Now, remember, radial symmetrical means that if they're round, basically, is what that means. The advantage of being radially symmetrical, you recall, is the fact that you're basically aware in every direction. If you're bilaterally symmetrical, remember, you're aware in the front more than the back. But if you're radially, however you're aware of the environment, however you're sensing the environment, you're on guard all the time. And when you want to move, if you do move, you may not move fast. You don't got to turn around. You just move in whatever direction you want to. All right, diploblastic, endoderm, and the ectoderm. Now, the endoderm becomes the digesting part, the gastrodermis, and the ectoderm becomes the epidermis, just like the sponge had an epidermis. What is important about this ectoderm, this epidermis, is there are stinging cells on the outside of that as well. So these stinging cells that they use to capture prey are also used defensively, because if this ectoderm right here, this epidermis, has the stinging cells in it, which it does, that's a mouthful if you want to eat that thing, right? You're going to get blasted with a bunch of these stinging cells that we're going to talk about. All right, we have tissues, but no organs. No organs. Remember, organs are the level right above tissue. So we're again there, Aristotle, excuse me, but we're kind of between the rungs of the ladder here a little bit, if you will. There's a group of nerve cells. They can tell touch and gravity, light receptors, And then a group of muscle cells that kind of works as contractile fibers, actually with contractile fibers. And that's what, when you see them when they move, bloop, bloop, that's how they do that. It's not really a nervous system. It's just a nerve net with some muscle cells. And now the reason for their name. The reason for their name is because they have a special cell known as a cnidocyte. There you go. And over on the right is a picture of one. And inside of that cnidocyte, which they use for, as I said, prey capture or defense, inside of that cnidocyte, there's a specialized organelle known as a nematocyst. And if you look on the right-hand side, a nematocyst is a coiled up thread, and it's sitting inside of that cell like a spring. And there's a trigger, and that triggers a physical thing, if you will. Now, when something brushes by that trigger, it trips that cnidocyst, and that nematocyst comes shooting out of that cell and sticking into its prey. See on the right-hand side, it might say cuticle of prey. So maybe that's a shrimp, right? Too close. Now that thread it injects, and not just one cell, however many cells you touch up against, that's what's going to happen to this. Shoots inside the body. And what you can't see in the picture here is that that nematocyst, that thread, actually has these little projections, not really hooks, that stick this way. So this thing really penetrates and holds on very well. In the meantime, they also inject a toxin through those cells, and that's where you get the stinging sensation. So when you go too close to a jellyfish, you get stung by a number of cells. Now, that can be deadly. Portuguese man of war can be that way. The sea wasps over in Australia can be that way. But you're going to get a stinging sensation from the ones here in Florida because you're a little too big for them to kill. So you're basically going to get a defensive kind of response. You know, they don't elicit it that way. You brush up against them and they shoot you with those nematocysts. But they're not going to reel you in, especially the ones you come into contact with and try to eat you. Isn't that cool? All of them have it, and it's unique. No other animals have anything like this. I think that's pretty clever. So that's the good thing. The bad news, you only have one gastrovascular opening, so mouth and ouch, waste discharge goes out the same place. So your mouth and your anus are the same thing, right? Much like some of the politicians that we know. But in that gastrovascular cavity, in spite of the fact you have to share your mouth and your anus, not the planet, it's very thin. So inside there, digestion can occur, and the beauty of that is digestion is kind of contained. You see in the pictures down below, the yellow there is the gastrodermis, okay? What we have here are the two types of cnidarians, the one that faces up, which is the polyp, and the one that faces down, which is the medusa. We talked about that with lobelia. We'll get to that. But look at the gastrodermis, okay? Inside of there, you can increase the efficiency of digestion because you're by phagocytosis. They just do it just like you learned in Bio 1. Because the food is contained in there. It's not really like the fungi, remember? They secreted enzymes on the outside of their bodies. And so while that made a nice soup of stuff to soak up, it's kind of wasteful. You know, it's a little more efficient if you can bring the food inside it. So that means less escapes, less gets out through the mouth/anus. You get a better return on your energy to capture that prey by bringing inside of that gastrodermis. The mesoglia that you see there is not a tissue layer. That is another one of those. That's the jelly of jellyfish that you feel. So it's not a tissue layer by any means. Now, as we saw with the lobelia, when it comes to the polyp and the medusa, and there's a picture, a sea anemone would be a polyp, and then the regular jellyfish is a medusa. Like we saw with the lobelia, though, that can happen in the same critter, right? Because remember, if you're sessile, and lobelia is a sessile, it is a polyp. It grows, you know, sitting still. Polyps grow up, right? In order to disperse, you might use the medusa. You remember when you looked under the microscope? There were two types of polyps, weren't there? The ones with the tentacles. Intentionally. Oh, do they have? Oh, good question, Patrick. Let me think. me see. Don't really have a nervous system like that. Don't have a probably not, unless there are some species that the trigger can be tripped chemically. So, for example, if there were a chemical around a prey item, some chemical that the prey item gave off, if that chemical somehow would trip the trigger, maybe. It's a good question. Look that up for me. We'll talk about that in class next time. Don't let me forget, because that's a good question. I never thought about that. Because I don't think they could do it nervously, but they might. Yeah, nice. Thank you. What the heck was that? Yeah, the obelia. You remember now with the feeding polyps and the reproductive polyps. Now the reproductive polyps made medusas. Now, don't get confused. There are medusa jellyfish, right, that spend their life as a medusa. This is a medusa that really is. It has just one job. It's a gonad medusa. It makes sperm and egg. As a matter of fact, the medusa, if you notice, are on the diploid side, right? And when they undergo meiosis, create all that variability we talked about, you get the sperm and the egg. So the medusa are only around long enough to produce the sperm and the egg. The obelia spends most of his life as this colonial polyp you see here on the left. So this is sexual reproduction. This is the larvae that's formed, by the way. This looks like it's like a jelly bean that's been under the fridge for a couple of days. You just thought of that. That's disgusting. Hydras can simply asexually bud, right? That's good, but genetic variation, you don't have any of that. There's the medusas of the obelia. There they are. Got some pictures of them. I like to find real pictures of these things. That's what those things would have looked like if you could have got them off that little polyp there. Old joke among biologists is gonorchism. Gonorchism means you're male or female. We're gonorchid. Jellyfish are very, very, very difficult to tell sex. So there's an old joke about jellyfish sex. Only they know the difference. They're the only ones who know males from males and females from males. I mean, that's probably a hyperbole, but nonetheless, it is very difficult to sex a jellyfish. So zygote, planktonic planula larvae, that's what that's called. That's the planula, the hairy jelly bean, into a polyp, and then the polyps move back into other polyps and medusas asexually, and then they produce the reproductive polyps. Coral is a polyp, okay? What coral does, the actual coral animal, you think, oh, that's that hard stuff. That's the exoskeleton of calcium carbonate, chalk. Coral is if you get up close to a coral head, a living coral head, you'll see the hard part. If you look close, you'll see the little tentacles. You can see the little tentacles of the anemones, or you can see the little mouths of the anemone. And people, of course, put their hands all over it, and that kills those organisms. As I said before, the big thing is now digesting on the inside. Extracellular digestion occurs, but on the inside, so that increases efficiency. Cooper's out in the back porch barking, so if he's bothering you, let me know and I can shut the door. Alrighty. Let's look at the different groups of cnidarians now. There's all kinds. So they're all going to basically be that gelatinous stinging. They're all going to have cnidocytes. There's the group that look the typical, the medusa. So we call those the medusazoa. Chironex fleckeri, that's the box jelly. That's a cubozoa. That's a type of medusazoa cnidarian. Very strong swimmers. Stings can be fatal to humans. In some cases, the species is more venomous than a cobra. Yeah, so death sometimes occurs within seconds. This is Australia, where all the weird critters are found. Give me 1 second to go get the dog to stop barking. Yeah, and Cooper gets out on the back porch and barks at something that walked through the grass 20 minutes ago. I'm like, it's gone. They're like having children. And I was going to say Australia is this weird place. I'd love to go there. My son's been there a couple times. His wife has actually lived there. She's from New Zealand. But, I mean, just the interesting life forms. You know, it's all that interesting adage that they say that there's millions of things in Australia that'll kill you. Most things you come across in this country are not going to kill you. But Australia tends to be a little different. And certainly the box jellies fit in that group. More venomous than a cobra. So that means it's neurotoxic. Neurotoxic. That's a good term. What does it mean if something is neurotoxic? How does it affect you? How does it kill you? Or how does it, kill you if you're prey? So if dead is dead. You got it. Yep. There are snakes that way, aren't there, Sean? Yep. Your snakes are not neurotoxic, though, are they? How do they kill their prey? Yep, they're constrictors. And then, of course, rattlesnakes, they're chemotoxic. And what they basically do is destroy tissue. That's why folks have been bitten by rattlesnakes very often are missing pieces of fingers. You go to any serpentarium. That'd be cool. Yeah, I think rattlesnakes are neat critters. Any serpentarium I've ever been to, the guy behind the glass who shows you how they milk venomous snakes, he's always, and I'm not trying to flip you all off, so sorry if I do, he's always missing pieces of his fingers. I've been working with rattlesnakes for 32 years. I'm like, yep, and you missed a couple of times, or they didn't miss. Who else? The hydras, cool. The Portuguese man of war. I went walking along Canaveral National Seashore one time, and there was hundreds of Portuguese man of war up on the beach. They get good size or small size. They don't. The big ones are the really problematic ones. What often happens, or not often, but sometimes what happens is scuba divers will. These tentacles can get 10 feet long. They won't see them very well, and they'll swim through them. And you get enough sting on this, you're not going to get eaten by the jellyfish, but you could die from all the stings. But what I want to say about the Portuguese man of war is it's a weird kind of critter too. It's what we call a siphonophore, and I put that in parentheses for you. Look what a siphonophore is. It's a step backwards again. It's almost into the Volvox colony of cells thing, right? Isn't that cool when you look at that? You always get those critters that are kind of living on the edge. So siphonophores, so what I mean by a colony of cells is there's a group of cells that make up the sail, the thing at the top. There's a group of cells that make up the tentacles, and then there's a group of cells that make up, I think it's the digestive parts, and they all decide to live together. I guess it's like one of those, hey, sail, you got no use without us tentacles. Well, hey, tentacles, you got no use without us gastrovascular cells. I don't know. It's neat. Yeah, I don't know. Kinda. They do, yeah. But it's, again, that kind of dog, wolf thing. Yeah, no kidding. Everything finds a way, man. The hydrozoas are the only ones that are freshwater. That's what I want to say too. They're the only ones that have freshwater species. That's a hydra up there with all of its. It can asexually bud more than once too. The jellyfish, the Scyphozoa. So these guys down here, those are the typical jellyfish. This one right here is a jellyfish we have here in Florida. This is known as Cassiopeia. There's an I in there. Opia. Oh, I think that's how you spell it. It's an upside down jellyfish. The tentacles actually, it's not a polyp. It's a medusa like the one on the right. We have a Indian River. I used to see them all the time. Those bright things in the middle, if you're looking from above, if you've ever looked at a jellyfish, nice circle, Steve. We're talking about the sort of bright semicircle regions. Those are the gonads. Tentacles hanging down below. But this one here, he lives upside down. That's just a different way of living. Most of these guys live as a medusa, but some of the ones in the coastal species might have a short polyp stage, and that may be sort of a resource partitioning thing. So not eating the same thing as the adults, kind of like the caterpillar and the butterfly that we talked about the other day, right? Of course, the big group of cnidarians, well, which one is not a polyp? The other species, you mean? Well, gosh, that's it's body structure. Let me go back to another slide. Jeez, Patrick, you're on fire today, dude. Let's see here. It must do with body structure. That technically, the body structure is a medusa. Because I always like polyps are up, medusa's down. But if the polyp would have a firmer base that would allow it maybe to stick to the whatever it's adhered to, the shape of the gastrovat. I think it's probably one of those things that if you took a Cassiopeia and flipped it over, it would look like a medusa. And it's not attached. They kind of float along the bottom a little bit. They can contract and kind of push themselves along the bottom. I hope that's good because that's my best off the top of my head. Yeah, that's a good question too. Yeah, I mean, there is a technical reason. Some cnidarian biologists would say, well, yes, because the blah, blah, the blah, blah, okay, thanks. But I'm sure it's something like that. They just look alike. And the big ones here, right? Sea anemones, but the corals. The sea fans. I had those big purple sea fans in class. We had them in lab the other day. Those were actually purchased at TJ Maxx. My wife brought them home and said, oh, look at that. Aren't these going to be pretty in the bedroom? I said, no, they're going to be pretty in my lab. But they're real. They're authentic, which is kind of a shame. But that's what a sea fan looks there in the picture, those big purple sea fans. They're filter feeders, right? Things floating through. Filter feeders with stinging cells so they can capture their prey. And the anemones, now in this group there are no medusa. It's just polyp. So what they're going to do when they reproduce is they'll produce gametes into the water, which will mix and then hopefully settle out somewhere. They can live by themselves or they can live in colonies. That's what a coral head is. So if you look down below, I have it labeled healthy and bleached, which I'll talk about in just a minute. But this is what we call brain coral for reasons that are quite obvious. And if you look very closely, if you can look closely down into that coral, each along those ridges are little tiny animals like you see in the picture right here. Little polyp cnidarians, right? Little corals, little animal. Coral's the animal. The exoskeleton is what we see in a coral reef. Okay, what's the whole healthy and bleached and what's the bad news on the left all about? See the percent coral cover? This is in the Caribbean, right? We only have one reef in the continental United States, and that's the one in Florida. So I'm not talking about Hawaii now, but as you can see, throughout the Caribbean, the percent of the coral cover is going down. It's dropping down. Now, coral provides beauty, tourism, lots of habitat for critters, diversity. I mean, I could go on and on, but let's talk about why they're dying, all right? If you look at that picture of what a coral is, you see the little brown spots? Those things are called zooanthellae. It's a dinoflagellate, a protozoan, a symbiotic dinoflagellate that photosynthesizes. So you get the connection, right? This is a symbiont. These dinoflagellates live symbiotically in the tissue of the coral. Now, the coral feeds, they're carnivorous, but the symbiotic dinoflagellate that photosynthesizes provides nutrients to the coral reef. As a matter of fact, in some corals, many species of corals, a lot or most of the food they get comes from what these zooanthellae do during photosynthesis and less from them capturing themselves. So they have their own little growing garden inside them. It's not a plant, but it is something that photosynthesizes. That's what gives coral the color, by the way. And the brain coral that you see down below, the greenish brown color, that's the zooanthellae that gives them the color. That's not the coral animal. The coral animals you see in the picture is clear. Now, there's something about corals that we haven't quite yet understood. When corals become stressed, and what I mean by that is the environment becomes something. Corals have very selective environments. They like it at a certain temperature. You know, you don't find them up north. They like it at a certain salinity, salt. They like the water of a certain clarity. They like it clear because obviously these photosynthesizers, if they're underwater, want clear water for the sun. But anyway, if any of those kinds of things change in the environment, the pH, the temperature, especially if it changes quickly, or if the environment ends up getting a lot of pollutants in it, in a stressed situation, the coral will sometimes expel, we say, the zooxanthellae. Get rid of them. Get them out of their tissue. Nobody's not really sure why. They think it might be a defense strategy gone awry, gone wrong, because unfortunately, as you can imagine, when they do that, they actually make it worse. Now, some corals can re-assimilate those dinoflagellates again. Some corals can take those zooxanthellae back in, and they can go on being alive. But in many cases, and especially down in the Caribbean, the corals either aren't able to do that, or they're so stressed that they're not able to do that, and they become bleached. The one on the right is a dead coral head. The corals are dead, you see. There's no color. There's no zooxanthellae. So the coral can't survive simply on just using its tentacles. It needs the zooxanthellae. And you see on the left-hand side, as from 1970 to 2010, I actually was involved right about here with a project when I was right out of grad school. I went down and I lived in the Keys. And I spent, a buddy of mine and I, or a fellow coworker, ended up spending all day diving all the coral reefs up and down the Keys and monitoring the bleaching and recording it. I used to get up in the morning and put on my bathing suit, my flip-flops, walk across the street, get into the lab, climb in the boat, and go scuba diving. That was my life. Unfortunately, it was a grant, and it was over too quick. But yes, what was going on is the amount of corals are dying off. Now, when the amount of corals die off, we lose an awful lot of habitat, right? The growing corals back, that's certainly something that's been getting some press. They grow really, 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 really slow. The University of Miami had a research boat down there. This is kind of an embarrassing story for them, but a number of years ago, at night, the research boat drifted on anchor and actually smashed into a coral reef. They anchored out away from the coral at night, but the anchor drift. Anchors can move. If you've ever been on a boat, you know. They weren't paying attention, and they mashed up a piece of the coral reef. And I think the estimates were somewhere in the vicinity of 250 years to repair the damage naturally, for the coral to grow back and repair the damage that was caused by that one ding with the boat. So that's the problem with that. And of course, if you think about where a lot of the corals are in the Florida Keys, and this is where I was, it was a lot of what I was doing in the study, and you'll excuse, this is Florida. The Keys go up this way. The corals are found on the straight side, not the Gulf side. Of course, Cuba's over here. But the pollutants that comes off of Miami, the nutrients, the fertilizers that come off all the sugarcane fields, they will cause algae blooms and perturbations, changes to the environment. So that's what's affecting a lot of the corals. It's what's causing them to bleach. If you haven't been down to the Keys to go snorkeling or scuba diving or anything like that, you must do that sometime. That's a really incredible experience, and one that you might not get to do forever. Much longer. I haven't been down there in years. I think I'm probably too disheartened to see the difference between the way it is now. They say most of the Great Barrier Reef has died off. My son said it was awful when he was diving there, just to see the way it... Humans, pollutants, and all that other kind of stuff. Okay, we got about 10 minutes. Perfect. I can talk about the flatworms, because those are cool. We looked at one of those under the microscope. But now we're into the bilateral critters, okay? And we're going to be into the critters that are known as the Lophotrochozoa. This group down here. So we're going to talk about these guys, the flatworms. So now we're into those, number one, thank goodness, right? Complete digestive system. Lophotrochozoa refers to two things. It refers to these group of critters called the Ectoprocta which have these feeding structures. Now, those aren't cilia. That's a very specialized feeding structure. Or they have a very distinctive larvae known as a trochophore. So if you have either one of those, or both, don't remember exactly, you're in the Lophotrochozoa group. This other group, remember, were the shedders, the ecdysis. Okay, so let's look at that. Let's do the platyhelminthes. Cool thing about science, folks, are the words. Platy means flat. Platypus, right? You go to the doctor and say, "Doc, I got worms." He might give you an antihelminthic. So platyhelminthes literally means flat worm, and you look at them, they are flat. There's one. There's planarian. He's cross-eyed too. They're all that way. Now, if you looked at the planarian in the sample jar, you could see that it was a little tiny brown thing, and you can imagine, I'm not going to let you handle it, obviously, because it could easily fall apart. Ciliated, that helps move it around. They're going to be wet, found all over the place, even on land, and there are even some land planarians here in Florida. Do I have a picture? Yeah, I have a picture of a couple of those. I'll show you later. No body cavity. Right, acoelomates, incomplete digestive system. Incomplete digestive system. I said amongst the bilateral, most have a complete digestive system, not all of them. The pharynx, can you find the pharynx in the picture? If this was under the slide, under a lab practical, could someone point out the pharynx to me? Emily. There you go. Thank you. Yeah, the pharynx masticates, chews, grinds up food. Okay. There are some parasitic species. Those are really cool. We'll look at those. These are the tapeworms. So if you love tapeworms, you're getting ready to really be fascinated. But first, before we do that, I want to talk about the platy part. Why be flat? There you go. You can draw me a square, a nice flat square. Why be flat? Well, here's why you be flat. It's all about surface area to volume ratio. You did the calculations in Bio 1. Don't worry. I'm not going to ask you to do the calculations, so I'm just showing you here. But you remember, the whole idea is to maximize the surface area to volume ratio of a cell. Now, that gets carried through out of the cellular level. It gets carried down into the microvilli in the lower right of your intestine. All those little fingers increase the surface area to volume. It gets carried out amongst the mycelial, the hyphae, the mycelial mat of the fungi. As it branches out, it increases the surface area to volume ratio, better transfer. It happens in the membranes of the thylakoid, folks. That's why those grana are stacked. More membranes, surface area to volume ratio. When you have a greater surface area to volume ratio, for example, 3-to-1 to 2-to-1, you're better at moving things in and out of your body or in and out of your cell. A flatworm doesn't have a cardiovascular system like we do. To exchange gases with its cells, it uses diffusion. It uses osmosis. It uses all those things that cells use. But it's an animal. So why be flat? You be flat for the same reason that cells stayed small. You maximize your surface area to volume ratio. And if you're flat like an ice cube tray, you see with the numbers, now if you want to really look at the numbers, you can. That's what I've done. I've taken something of the same volume and flattened it. You get a 3-to-1 ratio, just about. Right? So critters that don't have fancy cardiovascular systems, right, or fancy ways of moving water around their body or waste will very often use simple diffusion, but that limits you, just like with the mosses, remember? That's why mosses stay small, because they can't get big. You can do what? Right? It's all about that surface area. Of course, yeah. All about surface area to volume ratio, okay? That's why you be flat. Okay, let's talk about some clades. Remember, the clade is going to be a family group, so this has some taxonomic significance to it. So the two clades are the I always screw that word up, Catanulida and the Rhaptidophora. Two good tongue-twisting words. The Catanulida is a very small group of flatworms, just about 100 species, known as, for the picture on the right, as you can see quite clearly, chain worms. And they reproduce just by simply asexually by budding off. Right? So the offspring bud off and then they detach. Very simple. It's almost like a colony of cells, really, but it is an animal. The Rhabdidophora include the planarians, like you see in the picture. That's a Florida planarian, by the way. Notice where the picture came from, UF. I've seen these on my back porch, and they can get pretty good size, too. So if you look and you go, what a weird-looking worm with a hammerhead, that's not. That's a planarian. Get a picture of that. So planarians are free-living, and our buddy the tapeworm is not free-living. Tapeworms are parasitic, right? I got some cool pictures that we'll talk about tapeworms here. Yeah, in a couple of slides. I don't know if I'll get to those today, but let's look at the planarians first, because, oh God, I want to talk about their kidneys and then finish up. I got to talk about their kidneys. All right. Planarians, the free-living ones. Dugesia, that was the one we looked at under the microscope in the little jar. Was the genus is Dugesia. Kind of a cool word. No circulatory system, right? Diffusion and osmosis. Digestive system. Some digestion can occur outside of the body. Right, so there's some of that where they can secrete some of the fungi. The rest is inside the gastrovascular cavity. So they do a little bit of the fungus where they secrete the exozymes, but like the jellyfish, they found it more efficient to bring it inside. And when you looked under the microscope, you looked at maybe the dark little squiggly lines inside. That was the gastrovascular cavity. All right, now that gastrovascular cavity it emits the gastro part and the vascular part. The gases diffuse in and out of there, the water through osmosis, and the food diffuses from the pharynx into there. So that's the gastrovascular cavity is what delivers the nutrients to all the individual cells. Not really a brain, but a collection of nervous material, nervous cells at the front of the critter, which we call a ganglia. A ganglion is like a starter brain. Eye spots that only can tell light from dark. Back to our acceptations that we talked about in evolution. What good is 5% of an eye? There you go. There's 5% of an eye, and it's really good at telling light from dark. So you can know when predators swim over top of you. Most are hermaphroditic, and some of them you can split in half through fission and create other ones, so asexually reproducing. That's something rather common among ancestral critters, that you can do this kind of, just cut them in half. That's what you see in the picture. They sliced planarians in a lab, and they grow into two. Oh, perfect. 3 minutes to talk about flame cells. Their excretory system, their osmoregulatory system, getting rid of waste, balancing water, okay? The excretory system is something known as a protonephridia. Now, a doctor today who studies kidney disease is known as nephrologist. It's kind of the scientific word for kidneys, the nephrotic system, okay? Protonephridia means basically primitive kidneys. And it's these things called flame cells. So this is how it gets rid of waste material, like ammonia and that sort of stuff, and keeps the water in balance. So let me show you how it works. It's called a flame cell. All right, these flame cells, there's a flame cell right there. I get their name from inside of this cell. It's like a candle upside down, flame cell or flame bulb. So these cilia that are wrapped together beat back and forth like that. And what that does is it's physics, folks. This is where it all comes together with physics. That creates a negative pressure inside of that flame bulb, where that cap cell is. And as that does, water, see the black line, moves into there, and then collects in these tubes and out through the body. Simple. It's kind of like the same thing the Vorticella use their flagella or their cilia for, to move food around. So now we get a bunch of cilia, and cilia moves water. Oh, I can use it to swim. I can use it to move water. The interstitial fluid It's an acetylate, so these things are penetrating through this body cavity or this lack of body cavity, these innards, and it's those little cilia are moving back. And again, flame cells, because they look like a candle right back and forth. Is that cool or not? That is so cool how that works. It's so simple, but so efficient. Now, other critters, you stick with this. When we get into other groups, we'll see flame cells again. But before I leave you for the weekend, one last thought in mind. With Dugisia, metabolic wastes out through the mouth. All right, so it's kind of like the jellyfish in that case. So really cool in the flame cells. Still got a little work to do on getting rid of those metabolic wastes. All right, okay. Work on that, Dugisia. All right, folks, have a good weekend. I'll see you Monday where we will dissect the crayfish. All right. Talk about segmentation. Very much segmentation there.